Okay, a couple of announcements. So the light reactions lab report is due October 21st by 5 p.m. Just making sure you're aware of that. Keep that on your radar. The other, I've had a couple of students asking about the review paper assignment that you need to hand in coming up in a little while here. And I just wanted to spend a couple of minutes on that assignment. So review papers are covering a lot of information, right? And that's why they're review papers. So when we ask about, so the questions, if you pull up the questions and you look at these 5 questions that we've asked you to address. So I kind of went in order of from like start to finish of the review paper, in the sense that question number 1 is really thinking about the introduction. So you're reading the introduction, and that's where they're going to state what their objectives or the goal is for this paper, this review paper that they wrote. Same with question number 2. That is going to be in the introduction. So in the review paper introduction, they state, what is the big picture idea that they're going to talk about? Why is it important? Okay, so they're kind of justifying why they think that this paper is needed and you need to read it. All right, so question 1 and 2 are really coming from, sometimes you can find it in the abstract, and then for sure in the introduction. Question 1 and 2 you'll find if you read that section. It'll be there. Now question 3 is, I think, where a lot of you are like, oh my gosh, what do I say for each section of the review paper? And so if you look at, for example, this review paper, the mycorrhizal and abiotic stress, and you go down into the different sections. So there's your introduction, which is actually quite short, and then they get into the different topics. You're not expected to talk about everything they talk about in each section. For this assignment, that's not what we're asking. We'd like you to read the paper, but really you only have to pick out, if I were you and doing it, I would pick out like 2 things that you see in each section that seem to be worth mentioning, and explain a little bit about those 2 topics. So you don't have to go on and on and on and on. You can refer, you can also say, figure 1 gives a great example of this idea, whatever it is, okay? So that 3rd question is the bulk of the paper, where you're just picking out a couple of things from each section. Question 4, it may be that the authors don't really have some unanswered questions that they'll actually say, we have yet to learn blah, 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 or we still don't know blah, blah, blah. They may not say that. If not, is there anything that when you read it made you go, huh? And that's a very kind of subjective thing. It's whatever you think. So if the authors aren't explicitly stating anything to this effect, you can still probably come up with a question that you're curious about. So just answering that question, it's fine. If you give your opinion, then you get points for that. Okay, so don't get too hung up on that. And then question 5, that information of, do they really kind of sum up some of the most important conclusions? They usually have a section at the very end of the paper where they'll talk about conclusions or future studies, something like that, where you can pick that information out. Questions? Okay. All right. The other thing we're going to do today is review for the exam. So I'm going to lecture for a little bit, and then I will save time to review. So there's just a few last ideas on this topic of the light reactions that I wanted to mention, and then we'll start the next topic, which is in EDUCAT. That's posted on the carbon fixation. So that's the next topic. So here we're looking at this idea that when you look at the light reactions and you look at the two photosystems, they're both absorbing light energy. So for things to go really well for the plant, that needs to be done in concert with each other, in the sense that you need to have the same level of energy coming into both photosystems so that you can have a consistent and efficient flow of electrons from ultimately water as the donor all the way through to NADP+. But in reality, if you think about especially plants in a forested ecosystem where some leaves are getting shaded out, parts of the leaves are getting shaded out, some of the chloroplasts aren't getting their fair share of light, even within one chloroplast, they might not get equal light shining throughout that. So in general, they say it's probably pretty difficult to achieve in reality under natural conditions. So they've been studying that and looking at, okay, have plants evolved any kind of mechanism to deal with the messiness of real life and adequate energy input into both photosystem 2 and photosystem 1? And the answer is yes, they actually have discovered at least one protocol, one method that they've adapted to use under certain conditions. So LHC2, that is an antenna complex that you see, especially in plants that have adapted to shade or partial shade. They'll have beefed up antenna systems, so they gave them a special term. Light harvesting complex 2 is associated with photosystem 2 typically. All right. So what they find is when they've done experimental studies, if there's phosphate groups, so that little P means that there's a phosphate group attached to LHC2, what it actually does is it causes those antenna complexes, they can physically move through the membrane to give energy to photosystem 1. So there's actually movement where these antenna pigments can kind of go and surround photosystem 1 and allow for more energy to be put into photosystem 1. If that LHC2 experimentally, when they make sure that it's not phosphorylated, what happens is that it will keep giving its energy that it's gathering from the sun to photosystem 2. So it's actually able to move and deliver energy where needed, just on this very basic idea of whether or not it's phosphorylated or not. So that phosphate attachment is like the signal to tell the antenna complex what to do, where to go, where to deliver the energy. So it's quite amazing, but pretty simple. It's a pretty elegant solution. Now, when they've done this experimentally and they play with it in the lab, what they found out is that whether or not this whole situation happens, another key thing to think about is the plastohydroquinone levels. And so remember with plastohydroquinone, we see that plastoquinone is associated with photosystem 2. And pheophytin is in photosystem 2. It gives the electron to the first plastoquinone, which gives it to the second plastoquinone. and then all that repeats, and then that second plastoquinone gets a second electron, grabs 2 protons, and then it releases from photosystem 2, and it starts migrating through the membrane. It needs to get to cytochrome B6F. Well, if you have lots of PQH2, so this last bullet point says high PQH2 concentration, so the bracket means concentration. So if you have high concentrations of PQH2, it's actually another signal to the light harvesting complex to shift energy to photosystem 1. So what's happening here is let's go to a visual. If you have high PQH2, whoops, not that visual. Where is it? This visual. If you have high PQH2, that means right here, this is building up, it's building up, it's building up. And it's like, okay, what would cause PQH2 levels to build up? It's basically saying that somewhere downstream, there's something stalled out. And it can't pass its electron forward because these electrons, it's not getting passed forward. And so what this is saying is PQH2 is building up, something downstream, so to speak, is messed up. And what usually is the case is photosystem 1 is not getting enough light energy. And so photosystem 1's electron doesn't get excited. That means plastocyanine can't donate its electron. That means all of it bogs down and slows down. And that causes PQH2 to build up, because PQH2 is coming from the fact that photosystem 2 is getting hit with light energy, hit with light energy, hit with light energy. And so PQH2 gets made. But if photosystem 1 is not getting hit with the same amount of light energy, then it's going to all start to grind to a halt. As PQH2 releases, like this is fine. This initial part is, as long as PS2 is getting hit with enough light energy, you'll have PQH2 moving along. But if photosystem 1's not getting hit with the same amount of light energy, then things start to grind to a halt. Does that make sense?
Is there anything that's like
So far they haven't. They're looking, but so far as far as I know, they haven't deciphered that one. So there tends to be kind of lopsided focus in the research on photosystem 2. And I think it's primarily because they're still trying to figure out the oxygen evolving complex, how that all works. Still kind of a mystery. So there's a lot of effort and research on photosystem 2, and there's less focus on photosystem 1. Okay. So that was, I think, the last main concept I wanted to get across for what's going on with the light reactions. I've also posted into EduCat oh boy. Okay, let's go into this one. A short slide set has been posted on the summary of the light reaction. So what I've done is I've pulled the PowerPoints from the large light reaction slide set and put it into this slide set where it's just focusing on the light reaction process. All right. And so this might be useful for studying because it's kind of succinctly all put together in one slide set. So we've been talking about this for several days now, and this idea that you have the light energy striking both photosystems, hopefully at the same level of energy input, and the idea that you have ultimately, once this chlorophyll electron in the reaction center gets excited and boosted off to feel fighting, then you ultimately have the replacement electrons coming from water. So water is your ultimate electron donor. It will donate an electron through the actions of the oxygen evolving complex so that that chlorophyll molecule that just lost an electron can gain one back and then get hit with light energy and have that happen all over again. So this reaction pathway that you see on this slide is good to study because it gives some of the good detail of all the players. So this previous picture I just showed you leaves off, like you don't see theophyte in the next year. Okay, we talked about it, but it's not being shown here. But on this next slide, it is being shown. Okay, so if you're trying to piece it together as you work through it, I like this because it's putting things into context of where everything is happening and the major points, and then this fills in the details that we've been talking about. They also put this on an energy scale, so they're trying to show that once that reaction center gets some light energy, it really excites that electron. So they're trying to show how it goes up on the energy scale like that and allows for handoff of the electron field fighting, and then first PQ and the second PQ, then all of that has to repeat again so that that second PQ can get 2 electrons. And then it grabs those 2 protons, which is important. So they're trying to show here how there's 2 protons coming from the stroma. So you see protons disappearing from the stroma, free protons are disappearing. So PQH2 grabs those 2 protons, and then it can release off and travel through the membrane, bump into cytochrome B6F, and then it can give off those 2 electrons. So in the same slide set, we talk about that as well, where we get into the donation of those electrons. One goes the linear path, one goes into the Q cycle, and then the 2 protons get dumped into the lumen.
So, like that seems, like use that,
yeah.
like the free energy.
Yes, exactly. So this is trying to show the downhill idea, so to speak, the energetically downhill idea of electron transfer from donor to acceptor, and that provides a little bit of free energy to get those protons dumped, so to speak, into the lumen of the thylakoid membrane. So that's really important. And then, this electron starts going into the Q cycle. You have cytochrome B, and then the quinone, it's the same kind of quinone, the plastic quinone. So it gets that 1 electron, but again, it's going to need 1 more. It's like, I need 1 more, otherwise I'm not going to do anything. I need 1 more. And so all of that has to repeat all the way back from photosynthesis 2 getting hit and having that all happen again. And then another PQH comes along and says, okay, I'll give you that second electron that you need. And then that plastic quinone again grabs the proton from the stroma. Again, you see disappearance of free proton out of the stroma. And so that Q cycle is a way to develop a nice proton gradient. You do see that developing with the help of the Q cycle cooking along. It really does help boost the proton gradient idea. And then if the linear path, if you're thinking about that, you see the linear electron flow to iron sulfur instead of F, and then the heptocyanin mobile carrier. And then it jets along the lumen portion of the thylakoid membrane, hopefully gets to photosystem 1. And then you see that photosystem 1 has to get hit with light energy, got to have that, so that its electron in the reaction center can get excited, boosted off to A0. So we talked about that as well, and how we have in photosystem 1 these different players, A0, A1, iron sulfur X, A, B. So those are the 5 players in photosystem 1 that are passing the electron down the line, so to speak. And then finally, you see that the iron sulfur B, everything's going okay, ferredoxin will get that electron. And then once ferredoxin gets that electron, it can release from photosystem 1, and it's in the stroma. It needs to find that FNR molecule, because that FNR is really important. It's going to allow for NADP plus to get reduced. So FD and FNR are both mobile carriers in the stroma that will allow for ultimately NADP plus to get reduced. So by the time the light reactions are done, the final electron acceptor is NADP plus, gets reduced, NADPH, and then now that is going to become important to be used for the carbon fixation that we're going to talk about next. And then as that's all cooking along, you have that electron transport chain process, you get this proton gradient developing. They go hand in hand. And so if everything's going okay, you'll get a buildup of protons in the lumen. And so they'll diffuse back through that ATP synthase. And as they're diffusing back, it creates a situation that allows for the ADP to get phosphorylated to ATP. Okay, so that's what that term linear photophosphorylation means. It just means that as the electrons are going on down the line, you will get ADP phosphorylated eventually as the protons diffuse back through the ATP synthase. So this slide set kind of takes what we've been learning from the big slide set and puts it into, you know, I just kind of shortened it up for studying the process. Is there anything right now that's you have questions on with the light reactions? I know it's probably not your main focus right now because of the exam coming up. I get it. But having said that, after the exam on Friday, I really, really, really encourage you to turn your attention to the light reactions if you have not done so. So this is what, the third or fourth lecture that we've had on it. So the light reaction slide set, none of that will be on the exam. Yeah, yeah. So I guess it ends with biological nitrogen fixation. That was the topic right before. No, I decided not to. So the whole light reaction slide set will save. Yes. Yes. Yes.
So on the next exam of this, is there a potential like having a diagram that we have to with
Potentially. Yeah, there might be a labeling anatomy type thing. Huh? So yeah, I mean the third exam, of course, so I don't know, what is this, the fourth lecture period that we spent time on the light reactions? Obviously it's a big deal. If I'm spending that much time going over this, it's a big deal. So you bet the light reactions are going to be on it. This process, it's going to be on it. So that's why I get that you're maybe not focused on it right now, and I understand. However, I really encourage you, don't wait till the last minute to start learning this. You're going to get buried quickly you're drowning in information. It's too much to learn just in a couple days. Okay. And I really encourage you to think of creative ways to work with this material as you're learning it. I used to actually act it out, like I was acting out a play. It made sense in my head. I did it where no one could view me as I'm moving around my room, but that worked for me. Other people tell a story and they come up with some kind of story where they weave this all in to the story. And then all they have to do is remember the story to remember the process. So I encourage you to get creative because it's much easier to recall that information than this just trying to do rote memorization. Okay. I'm considering holding a review session for the third exam as we get closer to the third exam. I've done that in the past and some students have found that useful, so we can talk about that if that seems like a good idea for this semester. I usually do it in person. I find a room. We usually do it in the evening, sometime in the evening, something like that. Okay. Let me get into the next slide set on carbon fixation. We need to think a little bit about the idea of, okay, once the plants have done the light reactions and they make that ATP and they make that NADPH, so what? What's the big deal? So we're still going to zoom into the chloroplast. And now we're going to zoom in particular to just the stroma portion of the chloroplast. So the watery portion inside the chloroplast. If you were to crack it open and peer in there, that's where we're going to zoom in. So we just got done talking about the idea of NADPH and ATP getting made through the process of the light reactions. And then those are the products that are showing up in the stroma. So now you have to imagine that the Kelvin cycle, the C3 cycle, carbon fixation, depends on who you talk to, what they call it. But here you can see it's labeled Kelvin-Benson cycle. I tend to just say the Kelvin cycle to shorten it up. And so this is a picture showing you the link between the light reactions and the biochemical reactions. So you're seeing what we've just talked about in a very summary image here on the left portion. And then they're saying, okay, now once you have the ATP and NADPH made, that can immediately feed into the Kelvin cycle. And from that, the plant is grabbing CO2 from the atmosphere and through the Kelvin cycle, synthesizing carbohydrate. So CH2O at that end, that's the basic building block of a carbohydrate. So we're thinking about this big picture equation again that I have with the CO2 and the water reacting to get you a carbohydrate and oxygen. Okay. So we're coming into now, okay, now we finally get to talk about what happens. How does CO2 come into this Kelvin cycle and how do we end up with carbohydrate? So they call it the C3 cycle because the first stable molecule that they can detect in the Kelvin cycle is a 3-carbon molecule. So that's why it has the nickname C3 cycle. So sometimes I'll call it the C3 cycle. It's the same thing as the Kelvin cycle. When they say carbon fixation, what they're talking about is CO2 getting fixed into carbohydrate. So when you hear that phrase carbon fixation, it's CO2 to carbohydrate. So when they looked at the Kelvin cycle a few decades ago, when they were figuring this out, they could tell that there was a 3-carbon sugar showing up, and they could detect that with the technology at the time. They figured out that you have the ATP and the NADPH funneling in key points to the Kelvin cycle, and that those molecules are coming from the light reactions. But they had a heck of a time figuring out what does CO2 react with to get you that 3-carbon. So, we know CO2 is a 1-carbon molecule. So they kept looking for a 2-carbon molecule. They're like, okay, 1 + 2 = 3. So there's a 2-carbon molecule that CO2 is likely reacting with. And so they were hunting for this 2-carbon, couldn't find it. And so then Dr. Kelvin, his full name is Melvin Kelvin. I wonder how many times he got picked on for that. I don't know, especially in elementary school. But anyways, Melvin Kelvin, he did his undergraduate work at Michigan Tech University in the Keweenaw. So when I did my PhD work up there, he was talked about a lot because he went to school there. Anyways, Melvin Kelvin came along and he said, well, okay, there's more than one way to get to 3 carbons. Yes, maybe it's a 2-carbon molecule that CO2 is reacting with, but there's more than one way to get to 3. And so he kind of broke open the idea of let's look at it from a different viewpoint. And he actually said, what if it's a 5-carbon? And instead of a 2-carbon, and that CO2 reacts with a 5-carbon, well, that gets you 6 carbons. Then if you divide 6 by 2, you get 3. He says, that's another way to get to 3. And so they started looking for 5-carbon and they found it. And so it was RUBP. Full name is ribulose 1,5-bisphosphate. So there's 2 phosphate groups on it. Okay, 1,5 position. All right, so RUBP, you can just abbreviate that down to R, capital R, small u, capital B, capital P. All right, RUBP. So that's the 5-carbon sugar. And for that, he won the Nobel Prize, Dr. Kelvin. He won the Nobel Prize for his work on the C3 cycle. They named it after him. They said it should be called the Kelvin cycle. Then eventually they said, oh, Benson should be on there too. And in fact, I think there's a third one. Yeah, he's another scientist. And then sometimes I think they have a 3-scientist name to this cycle. I just shortened it down to Kelvin. So yeah, there's a third, I can't even remember who the third scientist is, sorry. So this is the big picture idea of what's happening during the Kelvin cycle. I refer to this as the pie chart diagram. So if you hear me say the pie chart diagram, this is what I'm talking about, all right? So they've broken it down into 3 stages. So the top right is where it kicks off, where you have the, wow. My laser pointer just stopped working for some reason. Here you see the CO2 coming in, all right? So here's the start. The stomata open, sun comes up, sun energy strikes the plant. The plant will open up its stomata, CO2 starts to come in, and hopefully there's plenty of RUBP available. So RUBP and carbon dioxide will react. That's called the carboxylation reaction. All right, so what they mean is RUBP is getting carboxylated. That, it's reacting with CO2. When that happens, there is a 6 carbon molecule that gets created, but it's really unstable. And back in the day when Kelvin and his associates were looking at that, the reason they kept looking for a 2 carbon is they never saw the 6 carbon unstable molecule. The technology wasn't good enough. And so there is a 6 carbon molecule that's there, but it's very ephemeral and quickly breaks down into 2 molecules of phosphoglycerate. So this is the first stable molecule, which is 3-phosphoglycerate. You can abbreviate that down to PGA. So another way to say phosphoglycerate is phosphoglyceric acid. So PGA, phosphoglyceric acid, is another way to say phosphoglycerate. Anytime you see ATE in science, that means it's an acid. Okay, so that blue portion of this pie chart is the carboxylation step. So here's what we just described. CO2 reacting with RUBP gives you an unstable 6 carbon that quickly breaks down into 2 molecules of 3-phosphoglyceric acid. The enzyme that facilitates that reaction is called Rubisco, which is an acronym. It stands for ribulose 1,5-bisphosphate carboxylase oxygenase. So it's a mouthful. That's why I'm glad they came up with the acronym Rubisco. So the C and the O on the end of that acronym, the C stands for carboxylase, the O oxygenase. So anytime you see ACE on the end of a word, it's an enzyme. So this is a funky enzyme because it can do 2 different things. It can carboxylate, which is what is being shown in the first bullet point, that's carboxylation, where RUBP is reacting with CO2. So that's all great. Unfortunately, it can also act as an oxygenase enzyme, which means instead of CO2, it will allow oxygen to come into its active site. And if oxygen reacts with RUBP, it's a different ballgame that we have to think about. We'll talk about that. But first, we're just going to focus on when Rubisco is acting as a carboxylase enzyme. This enzyme, by the way, is the most abundant protein on the planet. There's more organisms and more copies of Rubisco out there than any other enzyme. So when they calculate how much is actually showing up in leaf tissue, they say about 40% of typical leaves have that 40% of the protein is Rubisco. So it's a very abundant and very critical enzyme. If you like to see the chemistry of what's going on, so here on the left is RUBP reacting with CO2. So if you want to see that visual, I put that in for study purposes. I'm not going to ask you specifically what carbon CO2 gets bonded with. It's just a visual in case you like to think that way, okay? And then you see the 2 molecules of PGA. They're identical. 2 PGA. All right, so what we just described is the carboxylation step. Now, you see there's two other points to the pie chart. There's a reduction step and a regeneration step. And so you can see here what we just described. Nowhere in that conversation was AATP showing up or NADPH showing up. But you do see it in the next two phases. All right, so in the reduction phase and in the regeneration, both of those have ATP input. And then reduction also has to have NADPH. So now you're starting to see the link where the light reaction products are coming into play here. But they don't come into play until reduction regeneration. So what that's saying is the first step, as long as CO2 is available and RUBP is available, they will spontaneously react in the Rubisco active site. There doesn't have to be any ATP to push into it or any reducing agents like NADPH. It just will happen. And if everything's going okay, that's going to cook along all day long. But sometimes situations aren't perfect and the plant has to deal with Rubisco also taking oxygen in. And so we'll talk about that. But first we're going to get down the Calvin cycle. But I think I'm going to stop here for this material because I wanted to shift and leave some time to answer any questions you may have on the exam material for Friday. Let's see. So phloem translocation material. Let me pull up that slide set. Okay, so this is the phloem translocation slide set. So questions on this material?
To get it, is that like made by the to get it out, or is that like information that like makes it? Oh wait, no.
No, I think you're talking about.
Yeah.
So yeah, we'll get to that. Did you? Yeah. Yeah. That one, right? Okay. So, okay, we have a couple of things going on here to think about. Sieve tube element is being shown, companion cell, and then this is the source cell is like a leaf cell. And it's doing photosynthesis, making all that lovely sugar, and then it is dumping or loading that sugar into the sieve tube element. So you do have to think about water potential concepts as the loading and unloading is happening. So let's think about that. They even give you some numbers to look at if that helps. So if you dump sugar in to the sieve tube element, and you have to imagine lots of source cells doing that near that vein. So now you get a bunch of sugar building up. That's going to mess with the psi sub S value of the water potential equation because you're putting more and more solutes, psi sub S, more and more solutes. So it's going to drive your psi sub S to be more negative. Well, what's impact does that have? If your psi sub S is getting more and more negative, that's going to drive your overall water potential more and more negative. And it's going to be more negative than some surrounding cells. And here they've drawn it that it's more negative than the xylem water, the vessels or tracheids. And so they're showing how water is moving from a less negative water potential in the xylem to a more negative water potential in the source sieve tube element. Okay. All right, so you have water now because of all that sugar moving in, driving your psi sub S more negative, your overall water potential gets more negative than surrounding cells. So water rushes in. Now you've explained how psi sub P is starting to go up because you have turbo pressure developing in there now. So your psi sub P is going up, some positive number. Questions on that?
What is the water cross xylem and there's membrane barrier. No, that's not involved.
It crosses through the cell wall of the tracheids and vessels to the cell wall of the sieve tube element, and then gets passed through the, doesn't really, it can go through the membrane of the sieve tube element, but usually through aquaporins. So yeah, you won't be asked that. You don't need to know that level of detail. Just say water rushes in from somewhere else that's less negative to more negative. I think about the water potential. Why is water rushing? Because you have less negative water in the xylem, more negative water potential in the pool. So that's the loading idea, what's happening in the loading in the source. But that's half the story. You have to finish the story. So to finish the story, you talk about the sink, what's happening there. Because for sugar to move, it's based on a pressure gradient. So we've established that you get some positive pressure here, water moving in, that turbo pressure. But now you have to say how there's a gradient, a pressure gradient. So that is the second half of the story, where now, let's say it's a potato tuber tissue saying, I need more sugar to build my tuber. So now sugar is getting unloaded at the potato tuber. It's getting dumped into the actual tuber tissue. And so that means that the sucrose is leaving, exiting the sieve tube element at the sink end. That's going to make your size of that approach going to be less negative. It's going to get closer to zero. It's going to get less and less negative. That means it's going to drive, shift the water potential. So it's going to be less negative. So now the water has an opportunity to go from less negative in the sieve tube element to somewhere more negative, which is usually the vein right next door. I mean, the xylem right next door in the vein. All right, so that's why they're showing water leaving, following the laws of less negative to more negative overall water potential. Well, if you have a bunch of water exiting at the sink area, that's going to drop your size of P. You're not going to have very much pressure due to turbo pressure if water is exiting. Now you've completed the story. Now you can say, so now you have a pressure gradient. High pressure in the sieve tube element at the source, lower pressure in the sieve tube element at the sink. As long as that gradient is maintained, you'll have sugar dissolved in the water, moving from source to sink. Okay, so you have to tell the full story because you have to show how that gradient is created. And if I ask about it, you do have to think about water potential, like what's happening to size of S, what's happening to size of P, what's happening to the overall size of W.
So this correlate as the, I guess that sink cell would be the super kind of thing, like as the water sink because that
Yep. 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 Other questions on phloem translocation process? Yes. Yes, so allocation versus partitioning. In this class, they have two very particular meanings, definitions. So allocation is looking at, I'll put it in green, metabolic fate. If you can link allocation and metabolic fate, what is the plant going to do with that in terms of, should I store it up for a rainy day? Should I immediately use it for, whatever metabolic needs it has? Or should, if it's source tissue, it can, of course, hopefully take a portion of what it's making and send it off to a sink. So allocation is, how should I allocate all of this sugar? Should some go to storage, some go into translocation, some used for metabolic support to keep me alive? Okay, so the metabolic phase. And then partitioning is, now we're thinking about all the different sinks on the plant. And we're saying, where does the source decide to put, if it's going to translocate it, who gets it? So partitioning is thinking about partitioning off different parts of its body. Like I want 1/3 of it going into my roots. I want another third going into my flowers. So partitioning it into different parts of the body of the plant, if you want to think of it that way, maybe. And that the key thing too is that sinks are competitive. The sink tissue is communicating back to the source tissue, and some sinks are more demanding than others in terms of I need more sugar right now because I'm young, I can't do photosynthesis fast enough, or maybe I can't do photosynthesis at all, like a potato tuber. But I'm really actively growing. And so we talked about sink strength is the size of the sink and its activity. And so you factor both of those in to calculate out the sink strength. And so the different sinks are all vying for that sugar. And so the plant has to partition into those different sinks. Does that make sense? So I try to get you to not think about the common use of these words out in common everyday life. In this class, they have very particular meaning. Other questions? Two essay questions. So I don't think that's going to change. It's stuck at 2 right now. So let me pull up. I think your question might have been biological nitrogen fixation on the minerals. Like, oh, the phytosidrophores and all of that.
One last question.
Yes. Yes. So the polymer trapping model for sure. Yes, that was a rather brilliant hypothesis that they proposed that it was a conundrum at the time. It's like, how the heck, why doesn't the sugar just back right out and slow down its concentration gradient right out of the sieve development? So the polymer trapping is a way to get the sugar transported and trapped into the sieve development so it doesn't flow back out. So the polymer trapping model. Now, so the other thing at the sink, I didn't really spend much time talking about unloading options. I just said there's lots of different options. I kind of left it at that, so no detail on that. But some of the loading ideas we talked about in some detail with the polymer trapping. And so mineral nutrition covers, of course, the minerals. Remember I told you table 4.2 plus the PowerPoint that I have talking about minerals are any questions? And then the mycorrhizal fungi. I did say, you know, thinking when you're learning this material, a good way to learn it more deeply is compare and contrast. What's similar between the ectomycorrhizal fungi and the endomycorrhizal or VAM fungi? What's different? So I think that's a good way to study it. And then the minerals, table 4.2, and then some of the other, like, you know, the nitrogen, phosphorus, that information.
You want us to know which, like how the elements is up, like what, so how like nitrogen.
Probably the most important one is nitrogen. That nitrate is the preferred form. I think that really is the only one I would ask about in detail. All the other ones, it's like they need phosphorus. I'm not going to say what form. I'm not going to ask that. But nitrogen, we did talk about that because it's a problem in the real world, because if you over apply nitrogen into fields, it can be a real problem. You don't no, my iron. Okay. So the iron thing, you're good on it. Okay. What else? Yes. The terminology. Yeah, like I'm not gonna ask you to memorize like iron deficiency looks like this, whereas phosphorus deficiency looks like that. That I'm not gonna ask. Yes, the terms, like, so what does chlorosis mean versus necrosis?
But we don't need to like put that.
To a particular element, you mean?
Or, yeah, or like,
You might get a visual and say, is this chlorosis? Is it mottled chlorosis? Is it intravenal? You might get some kind of visual, but I wouldn't say, what kind of deficiency does iron provoke? You know, I wouldn't do that, because, but just the terminology that you're comfortable and you have the general idea of what it would look like. Other questions on this or biological? Nitrogen fixation. So the nitrogen fixation, you know, that's also a process we talked about, that it's kind of a big story, right? You got the plant and the microbe, they got to find each other, and then they go through that step-by-step process. Okay, I will be checking my email regularly between now and Friday. If something comes up, send me an email and I will get back to you.
I know we made an appointment